your study guide when we get done. And that will be chapter 20. So we're going to work on chapter 20 today, but we will not do all of it. All right. So I know we've talked about this before. We talked about it at the beginning of the unit. But we're going to come back to it again. We talked a little bit about recombinant DNA before, and I didn't spend too much time on it. I did not test you on it. This unit I will. So I want to talk about it a little bit more because it is becoming very standard practice in science that may or may not be okay. That's another thing that we will talk about is if it's okay to do what we're doing. Kind of the initial question is, how does it work? And how is it? Because we can do this. I'll set it up as that. We can take human DNA and we can insert it. We can stick it into a bacterium and combine it with that bacterial DNA. And the bacteria will produce whatever protein that human DNA coded for. So we can make bacteria produce human proteins. Okay. And one of the questions is, why does that happen, or why is that possible? And the answer to that question is because our code is universal. Every living thing from a bacteria all the way up through us uses the same codon chart, has the same nitrogenous bases, A, C, T, and G, same amino acids. So that is kind of the long and short version of it. Now, one of the things we'll talk about a little bit later is the idea that prokaryotes don't have any what? That was way too ambiguous. Do they edit their DNA? Prokaryotes, bacteria. No, because they do not have a nucleus, but they also do not have introns. Everything that is in that code that is transcribed will be translated. Okay, so there are no introns in prokaryotes. Now that is going to be important later when we start talking about recombinant DNA, because that means that we have to be careful if we're going to edit our DNA, because if we take human DNA, a human gene, and we put it into a bacteria and we assume that it's going to be translated properly, we're in for a big surprise, because human DNA needs to be edited, right? We need to take out those introns, but we can't take out those introns in bacteria. So we've got to make sure that we do that editing before we put that DNA in there. But otherwise, the code is the same. Now, why is that a big deal? Well, first of all, it allows us to do all these cray-cray stuff that, at one point would have bordered on science fiction, or would have been science fiction. But it also gives a very strong piece of evidence for a single origin of living creatures. So our penultimate unit is going to be on evolution. Our next unit is going to be on evolution. And we'll talk about different pieces of evidence. But the fact that we all have that same nucleotide sequence, A, C, T, and G, the fact that we all use the same amino acids, that gives a pretty strong support for the idea that we did have all a common answer, or a common place that we came from. So in order to let's talk a little bit about some of the things that we use when we're talking about biotech, and we talk about recombinant DNA, we're really talking about genetically modified organisms, or GMOs. And you hear this word all the time. And some people are like, we shouldn't have GMOs. We should not tinker with nature. We shouldn't take the DNA from one species and put it into another. One of the things that happens in science often is we get technology that maybe we shouldn't be using. I mean, depending on your point of view. And so we start using this, and our ethics kind of lag behind. And so is it ethical to do this? And there's been cases where there have been GMOs that have significantly affected the food chain. And so you release this GMO out into nature, and it can do harm to the other things out there, the naturally occurring organisms. And so there's this, idea that we know how to do this um, long before there's any ethics to kind of deal with it. So it's just something to keep in mind. But anyway, in terms of genetically modified organisms, it's done in plants, really done in plants a lot, to produce new proteins of all sorts. There's this stuff called BT corn, which it produces a toxin, a bacterial toxin. So this is where we've taken bacterial DNA and we've put it into a plant, and that bacterial toxin kills a little pest here, this little corn borer or caterpillar that eats on corn leaves and kills the corn. This one's kind of a fun one. Fish berries. Fish berries are fish that have gotten a gene from strawberries. These are flounders specifically. Flounders live in pretty cold temperatures anyway, but now they are extending. I said that the other way around. It's a gene from flounders. Flounders live in very extreme cold temperatures, very close to freezing. So they call it the, quote, antifreeze gene that keeps them able to do that and function that way. And they've been able to put those into strawberries, produce fish berries that now have extended seasons, so they can grow into September, October kind of thing. Yeah.
Could you hypothetically put that gene into humans?
Yeah, you could.
Damn.
Yeah.
So we could be
it'd be much
how
more difficult, but yeah, then there's the ethics around that, right? I mean, do we do that? And who controls it? Does the lab control it? Does Abbott Labs or Baxter Lab control it? Right? Does the government control it? Yeah.
So it doesn't have to be when the cell is first developing. They can just add it in any kind of life cycle.
I don't know the specifics about it. In some cases, it is only in certain parts of the life cycle. Yeah, you wouldn't have to do it from initiation of the living organism, from insemination or from fertilization or whatever. Golden rice is another great example. Golden rice is kind of a yellow color because it's rice that has been enriched with vitamin A. And I'm not sure where that comes from, but it comes from another organism. And a lot of places around the world, they grow rice, but the rice is a cheap, easy, sustainable food. However, it doesn't have a lot of nutrients in it. So you can put vitamin A into that and increase the value, the nutritional value of that food. So there are good reasons for doing this stuff. Yeah. What's that? In theory, you could. Yeah. If you could find something that produces that, a gene that causes the production of that particular vitamin. Yeah. Okay, so how does this all happen? Well, I'm going to come back to a topic that we've talked about once already, and this is for Nick, I will talk about gel electrophoresis. I'm going to do that on Monday because I want this to kind of sink in before I do that. And I think after the weekend, that will be helpful. So gel electrophoresis is kind of a way of identifying whose DNA is whose. And I wanted to talk a little bit more about how we actually do that. We start off with these things called restriction enzymes. And restriction enzymes were originally found in bacteria. And bacteria all the time are subject to infections from viruses. So viruses will actually inject their DNA into a bacteria and trick the bacteria into making more viruses. And then the bacteria explodes, and now all these viral particles will be produced, and they can go off and infect more bacteria. So bacteria have developed a way to keep that from happening, and they're generally called restriction enzymes, meaning that these are enzymes that will look for a particular sequence in the DNA. And if they find that sequence, it's a sequence that does not occur in the bacterial genome. Okay, so in their code, in their genome, there is not that particular sequence. But if they find it in some other piece of DNA, they know it's got to be foreign. So if it's foreign, they can cut it up. And so they take an enzyme. If they find that particular code, they'll cut it. They'll break the DNA in half, and in doing that, will render it harmless, because if it's in chunks now, it can't work. So what we've done is we've taken those restriction enzymes and we've applied them to our own benefit, and we've used it for all sorts of things. It can be used in forensics, so CSI types of criminal investigations, that kind of thing. Medical, on Monday when we talk about Valerie and whether or not she has breast cancer. Paternity, who's the father. We've used it a lot in evolutionary relationships to see how closely related two species are. They may look a lot alike, but it turns out that when you look at their DNA, they're actually pretty far apart from each other. So there's lots of reasons to use this technology. And these slides you don't have, and you can choose to make a picture of them or write them down or whatever you want. But I wanted to look a little closer at restriction enzymes because I think it will help you a little bit with this whole process. So we're going to start with an enzyme a restriction enzyme that's looking for this sequence CCGTAT. Okay, and I just made up a scenario. I got two suspects. Do you see that sequence in there somewhere?
Yes.
Yes, I think that sequence is in there. I'm hoping it is in there. I'm hoping it's there. Okay, so I see CCG and then TAT. So where that DNA will get cut is actually right here. Yeah, I just draw a line. It's going to get cut. It's like, oh, nope, it's going to cut it in half, and half of it, or not half of it, but there'll now be two chunks of DNA. And I counted up the bases before, and this total chunk is 27 base pairs long. Okay, I could put BP. That is a shorthand that you see a lot of times in this kind of field. And this guy, I think, is 7 base pairs. And this one is 20 base pairs. Okay. So that's what happens when you take this particular suspect and you cut their DNA with this particular restriction enzyme. Okay. Now, we got another suspect. Suspect 2. And I didn't put the sequence, did I? Was it CCG? I think it was. CCG TAT. Do you see that in there?
Yeah.
It's kind of hard.
Yes.
Yes?
Yes.
Where is it?
Like next to the AUG.
Next to the AUG.
Yeah, there's a T.
Oh, there's a T there. Oh.
No,
Or is it the N?
no, I'm saying CCGTAT. That's literally like next to the two, next to the.
Where am I looking?
It's next to the first C, to the right of the first C.
I know there's two T's.
CCGTA.
It's TTAT.
It doesn't.
So what do we say? We say there was
No.
CCGTAT.
I literally see it. Like,
I know, but there's two T's. That is T.
oh, I'm looking at the bottom one.
Oh, that's because I didn't change the bottom one. So he's right.
What?
Don't be
I
hating
couldn't
on
see
him
the top one.
because I had changed it.
I always yeah, I never doubted it. I truly was like, he's about to
I don't know. I don't know what it was exactly. Let's just make it up. There. Okay. So this sequence was supposed to not be there, and I hopefully fixed it now, so it's not there. So CCGTAT is not in that first, the second suspect, right? We agree with that? No? Oop. Which means that we're going to end up with, instead of 2 fragments, we're going to end up with one that's going to be how many base pairs long? 27. Yeah. So then what we can do, and I had to draw this because I just figured it would be the easiest way, is we would load this into a gel. And so I'd have my, oh boy, I'm going to try this. Let's see how this works. Maybe. There we go. And on the side, I would have, the base pairs, number of base pairs. And let's just say suspect 1, suspect 2, and then we'll do a CS for crime scene. Okay. We would load our gel with our DNA samples. And then we would run it like we did in class, and we would see how fast the DNA travels down the gel. And what you should see, suspect 1, I said, was what, 7 and 20? Okay, 7 and 20. So are those bigger or smaller than suspect 2? They're both smaller. Which one's the smallest? 7. So 7's the smallest. So if I'm doing suspect 1, which one of their fragments is going to travel the farthest? The 7, because it's the lightest. Okay, so this would be 7 base pairs here. And up here would be my 20. Okay, I just made that up. No worries, freshman. Suspect 2. Suspect 2 has said 27 base pairs. So where's that one going to be in comparison to 20 and 7? It's going to be higher. Yeah, it's going to be up here somewhere. Okay. So that's how I know suspect 1 from suspect 2 if I use that particular restriction enzyme to cut the DNA. And they're much more sophisticated than what I'm showing you. They would have lots of cuts and they would have lots of enzymes. And then they could compare it to the crime scene. And, maybe the crime scene ended up being, I don't know, something like that and like that. So, did suspect 1 do it? They don't line up. Okay. If you're using the same restriction enzyme to cut the crime scene DNA with, it should line up. So they don't line up. And do these line up? Well, there's 2 fragments here, and there's only 1 here in S2. So they don't. So none of them. So both of them in this case would be exonerated, right, based on that DNA evidence. So that's kind of how that whole gel electrophoresis thing works. My computer's got a. Seriously? You're gonna do? Oh wait, seriously? Wow, it's slow today.
No, done. Done.
There we go.
No, done.
No, we're not done. Sorry. Okay, so these next slides are kind of that whole idea. So this is a much more sophisticated gel. And usually I said that on the side there were base pairs and, they would count how many base pairs you had. Usually it's what they call a standard ladder. And so that will have all of the different standard size fragments. So you can kind of decide what fragment, if one standard fragment is close to another and that standard fragment is 20, then then anything that's close to it must also be 20. But that's the whole idea of gel electrophoresis. Remember that the reason this stuff is traveling down the gel in the first place is the fact that DNA is very negatively charged. It's repelled by the negative electrical signal that's going through the gel, traveling through the gel into the positive pole. So here's another one where I've got some defendant blood, and then I've got blood from the defendant's clothing, from his jeans, from his shirt, and then from the victim. This doesn't look so good for the defendant, does it? Why not?
He's got victim blood.
Yeah, it looks like he has victim blood here. And just compare it. It looks like this is the victim here, and this is his shirt. They just did different concentrations of his shirt. That doesn't look very good. Looks like he's even got a little bit on his jeans too, maybe. Even here. He could have also been the one who came to the person when they were dying, and maybe that's why he's got it on him. So who knows? So it can be used in crime scenes. There's a few scenarios here that I'm not going to go over just because we did one already. paternity case as well. And then here's the cathartic part of today's lecture. You get to take out some stuff. Okay, so 20.3.
No, they're already X'd out.
They're already X'd out? Oh, well, do you want to X them out anyway? Will that make you feel better? I see. The thing is, I just never know if this stuff's going to come back or not, and I don't want to get rid of it. And, yeah. Yeah, I should have. What I should have done is I should have actually taken it out of your notes before I gave it to you. Sorry about that. Okay, so if we have a gene that we're interested in, like a human gene, for example, and we would like to use that gene to produce a protein, but not in a human being, we would like another organism to do it for us, we can do what is called gene cloning or DNA cloning. And we can do that with our bacteria because bacteria are actually really easy to work with. They reproduce very quickly. If you're careful with them, it's not an issue working with bacteria. And bacteria have a couple of features that are important. One is that in addition to their DNA, their kind of main DNA, they have these small pieces of DNA that are circular called plasmids. And they replicate, they copy the DNA independently of the other general chromosome that the bacteria will have. And what researchers can do is they can take a DNA molecule from a human being, a gene from a human being, and they can insert that, they can trick the bacteria or talk the bacteria or whatever you want to say. They can get the bacteria to accept the human gene and to put it in that circular plasmid DNA. And then once you have that, bacteria reproduce very, very quickly. So very quickly and easily, you can get many, many copies of the bacteria and also of the plasmid containing your human gene. So you've, in a sense, made many, many copies of that DNA molecule through the bacteria, and you didn't have to use human beings to do that. Now, I guess one of the questions would be, why would you want to do that? So I'm going to skip over this for just a second. I'll come back to it in just a minute. But again, some reasons for doing that could be that you want to insert a gene into plants so that it becomes pesticide resistant. That's one of the ones when I talked about how it's had devastating effects on the ecosystem. This one is broken bad in some ecosystems where they did it to control bugs that were eating the corn, so to make the plants produce more grain. But what they didn't realize is that it actually killed the butterflies that were eating the caterpillar version, or the caterpillar bugs, or an insect that was negatively affected by the plant. And then that had effects on the butterfly, which then had effects on the birds that ate the butterflies. So it really affected the food chain negatively. Another one that I have not seen a downside to, as long as it's done effectively, would be this one here. And this is why I went to this slide to begin with, was human growth hormone. So most of us don't have any problem hitting our predicted height. Might not be where we want it to be, but we get to it. And that's because of many factors, but one of them is what's called human growth hormone. And in some individuals, that human growth hormone is not produced. For whatever reason, it's not manufactured. And so you get these kids that have hit puberty and their growth is stunted. They're not growing at the normal. They're not hitting their marks, and they probably won't hit their marks. And what we've been able to do is we've been able to take other human growth hormone genes that that produce the right amount of hormone and cause the person to be of normal height, and we can insert that into a bacteria, and now the bacteria makes a lot of this human growth hormone. which then can be taken by the person who doesn't have the human growth hormone to get them to hit those marks and hit the height that they're supposed to hit. It's also used in insulin. So people who are diabetic rely on insulin. We talked about how insulin triggers the bringing in of glucose from the bloodstream. It used to be back in the day that the only way to do this was when cattle were taken to slaughter, for lack of a better term, they would take out the pancreas, which produces insulin, and then they would purify the insulin, and then that insulin would be given to humans, to people who are diabetic. And we've really seen recently that maybe it's not a good idea to be using another animal's anything for stuff like that to be put into our bodies. With all COVID, and we think that the virus jumped from a species probably originating in China somewhere to humans. And the same thing could happen if you're using insulin from the pancreas of cattle. It's possible that could jump. If there was a virus that infected them, it could jump to us. Max. And then
this is about the growth hormone stuff.
Mhm.
If like an adult took growth hormones, would they still grow or not?
I think they would.
Really?
Yeah. Yeah, I think it would.
Why people do leg extensions?
Wouldn't it make you get taller?
Because that's pretty tightly regulated, and it has other. I don't think that's. There's probably other health concerns wrapped around that too. You have to be very careful with it, and parents don't do that lightly. They don't just say, oh, you want to be a little bit taller. Let's. Doctors don't do that. They're like, it's only after they hit certain tests and don't hit certain marks. Miriam, do you have a question?
Oh yeah, I was just wondering, couldn't you think about viruses be true for bacteria too? It's like you were saying earlier, if a virus inserted DNA into a plasma, and then we use the same plasma to produce a protein.
Yeah, you're right. Yeah, it that I know of, that's never been an issue. I don't know if viruses are just a little more wily than bacteria are, but it's a good question, and it's a good comment to it's always good to check yourself on this kind of stuff because it isn't, quote, natural. It's nature, but it's taking nature in a different way than what it was intended. So we don't know what the consequences are. Yeah. All right, so I'm gonna run through this quickly, and then we'll run through it more slowly on Monday. There we are. Let me just go to the next picture because that one's. So the way this works is you take the bacteria that you're interested in, and that's usually E. coli. And if everything works, we will actually do this not next week, but the week after. We will be using E. coli, and we will be inserting a gene that was cut with restriction enzymes from a jellyfish that makes the jellyfish fluoresce. So you put a UV light over the top of it, and it'll be fluorescent. So it'll make the bacteria glow, basically. So you start off with the bacterium that you're going to use, in this case, our E. coli. And they have this little circular DNA called plasmid. It doesn't have too many genes on it. I think it's like 30 to 60 genes, whereas the whole bacteria, the bigger bacterial chromosome might have, I don't know, a few hundred. And you are going to cut that plasmid so that it opens up. And you're going to cut it with a restriction enzyme. And you're going to cut the gene that you are interested in with the same restriction enzyme. And when you do that, you are able to put that DNA from that organism, let's say that it's jellyfish, you're able to put that into the plasmid. And then after you have done that, you have we call it a recombinant bacterium that now has human DNA and its own DNA. And you can then let that grow, and it will produce the gene that you're interested in. In our case, it will produce the jellyfish DNA. It could produce human growth hormone. It could produce insulin, a toxin that will kill off pests, etc. A plasmid used to clone a foreign genes called a cloning vector. Bacterial plasmids are widely used as cloning vectors because they're readily obtained, easily manipulated, and easy to introduce into bacterial cells. And once you get them in the bacteria, they multiply very quickly. I do a lot of bacterial studies in here with different students because you can get results in a week, a couple weeks, because they reproduce every 20 minutes. As long as you are using sterile technique, which I teach my students, it's really easy to do, and you don't have to worry about issues. Gene cloning is really what we're talking about. You're making recombinant DNA, but you're doing it so you can clone genes. You can make more copies of them, and you can use that for all sorts of things. You could use it to produce a protein that is going to be used for research, and that's done extensively, as well as for other more health-related reasons for hopefully the greater good. I think, yeah. So I'm going to stop there. I was 3 minutes early.